Welcome to the Medical Cannabis for Clinicians Cannabis Growth, Manufacturing, and Testing Learning Module, Section 1, Cannabis Plant Basics. The main components of the cannabis plant are the seeds, roots, stems, fan leaves, flowers, and trichomes. It is important to understand that the majority of the cannabinoids produced by the cannabis plant are found in the flowers and trichomes of the plant. Virtually no cannabinoids are produced in the seeds, roots, or stems of the plant. Cannabis seeds, additionally, contain about 35% protein and fatty acids, which is why they have been utilized as a nutritional supplement. Cannabis stems grow erect, furrowed, and are often hollow. The average cannabis stem is 1 to 3 meters in length, but this can vary depending on the varietal of cannabis plant being grown. The stems are pubescent, meaning they are covered in downy hairs, and contain distinct nodes with alternating branches varying in length off of either side of the stem. The fan leaves are one of the most recognizable components of the cannabis plant. They are comprised of lanceolate leaflets with a central midvein, serrated margins, and an acuminate or pointed apex. The fan leaves average 2 to 7 centimeters from apex to petiole and are typically uneven in size, with the largest found in the center of the blade. The bottom surface of the fan leaves are typically whitish green, furrowed, and contain scattered glandular trichomes. The top of the fan leaves are typically dark green in color and contain rigid, curved trichomes. The male flowers of the cannabis plant are yellowish-green in color and consist of 2 to 4 millimeter pedicels terminating in 2.5 to 4 millimeter ovate sepals. Sepals consist of a filament and anther, which contains the plant's pollen. The female flowers of the cannabis plant can vary widely in color, from green to purple and red. The colas of the flower are the cluster of buds formed along the terminal end of the main stem as well as on ancillary large branches. Colas can vary in size upward of 24 inches when grown in a controlled setting. The colas are comprised of multiple calyxes and sugar leaves. The bract, also known as the calyx, is a furled, specialized leaf within the flower which houses the ovary. The bracts are covered in stalked glandular trichomes, which give the flower a frosted appearance. Each bract also typically contains 2 long, hair-like structures known as stigmas. The function of the stigma is to trap passing pollen to fertilize the plants. They can be white, pink, or yellow-green in color and up to 20 millimeters long. The stigmas will turn amber or brown in color as the plant processes through its flowering stage. The bract is also covered in trichomes. These are sticky organelles that are typically stalked in nature and contain terpenes and high concentrations of cannabinoids. Surrounding the bract are sugar leaves, which are leaves within the flowering portion of the plant, so called because they are covered in trichomes, which give them the appearance of being dipped in sugar. The pharmaceutical ingredients found in cannabis, known as cannabinoids, are found in the upper leaves, flower heads, and flower bracts of the unfertilized female plant. There are no cannabinoids present in the root, seeds, or stem of the plant. Cannabinoids are encased in tiny glandular trichomes located on the leaves of the plant. Some trichomes are large enough to be seen by the naked eye, but many are very tiny and give the plant a frosted, dew-like appearance. These trichomes manufacture and contain the cannabinoids. Some are bulbous, stalked, and glandular, while others have calcium carbonate crystals at the base and form a hooked, rigid appearance on the surface of the leaves. The sticky, cannabinoid-rich substance produced by the trichomes is commonly referred to as cannabis resin. Aside from its morphology, cannabis plants can also be characterized through organoleptic measures. Cannabis plants are incredibly aromatic and produce a wide variety of scent profiles dependent on the different terpenes and cannabinoids present within the plant. Common nomenclature for cultivars often include references to scent, such as skunk, mango, citrus, lemon, blueberry, pine, or sour. The cannabis plant also varies quite a bit in color. The color of the plant is influenced by the cultivar, the cultivation process, and the harvesting and curing process used for the plant. There's a wide variance in color from light or dark green to purple or even yellow-brown, and outdoor-grown cannabis plants are typically lighter in color than indoor-grown cannabis plants. It is important to note that regardless of the color of the plant, it should be consistent throughout the sample, with no gray, black, or white present, which can indicate the presence of mold or fungus. Cannabis also has a recognizable texture, as plants are sticky and resinous due to the high amounts of cannabis resin found within the trichomes of the plant. The taste of cannabis is typically bitter and sometimes acrid. Cannabis has varying commercial uses. The fiber types of cannabis plants contain low concentrations of cannabinoids and grow well in subtropical and temperate climates. China is currently the largest grower and exporter of hemp. The drug types of cannabis contain higher concentrations of cannabinoids and typically grow best in semi-tropical zones. In Canada, most United States, and Northern Europe, greenhouse cultivation is common due to a suboptimal growing climate. Additionally, large-scale eradication efforts in the United States during the height of cannabis prohibition led to increased indoor growing. Cannabis cultivation was reported in 135 countries to the United Nations Office of Drug Control from 2010 to 2015. North America is the largest self-supplying market for herbal cannabis. Europe is the largest consumer market for cannabis resin, which historically has been primarily supplied by Morocco. Cannabis sources among countries vary from illegal growth and importation to nationally controlled cultivation, such as found in the Netherlands. There are three primary sources of cultivated cannabis in the United States: federally legal material, state-regulated material, and illegally traded material.